So I've been talking a lot about PPAR alpha in the last couple of videos, and the reason is that, as this paper states in the abstract, peroxisome proliferator activated receptor alpha, PPAR alpha, controls fatty acid metabolism, and that sounds pretty important, and I don't believe it, but this is the kind of quote that you see in the literature a lot, and one of the reasons that I don't believe it is, so these are mice. These are PPAR alpha knockout mice. So that's this column here with the KO. So they don't have the gene PPAR alpha at all. And this is soleus. It's a muscle. And this is how much fat they oxidize in their soleus muscle. This is a normal mouse. They oxidize 151. And this is a knockout mouse with no PPAR alpha, and they oxidize 160. So it's weird that if PPAR alpha controls fat metabolism, that the mouse that doesn't have PPAR alpha oxidizes just as much fat as the mouse that does. I've introduced more reasons why I'm interested in PPAR alpha in the last few videos, but here's yet another one. So this paper, they used an inhibitor of something called the aryl hydrocarbon receptor, and it says AHR inhibition prevents and reverses obesity by likely reducing liver expression of PPAR alpha target genes. This is the same paper. They present this nice little map, and so here's the aryl hydrocarbon receptor. It's at the top of the totem pole, and it's activating PPAR alpha, and PPAR alpha is going ahead and activating SCD1, which leads to obesity and fatty liver disease, SPP1, which leads to obesity and inflammation, and other PPAR alpha regulated genes leading to insulin resistance and fatty liver disease. This is a bit of a conundrum if you think that PPAR alpha controls fat metabolism and that increasing PPAR alpha allows us to burn more fat. It's confusing why, if you prevent the upregulation of PPAR alpha using a pharmaceutical, why that reverses obesity. This paper is from 1962, and this drug is better known today as clofibrate. Way back in 1962, they said that clofibrate has been demonstrated to lower the level of cholesterol and lipids in serum and liver in rats. And they went on to show that oral administration of clofibrate was hypocholesterolemic in man, and that means lowers cholesterol. They didn't know it at the time, but the reason that clofibrate works is that it activates PPAR alpha, the supposed master regulator of lipid metabolism. They went on to do a large-scale controlled trial to try to show that clofibrate would reduce cholesterol and reduce heart attack deaths in men. Michael Oliver sums it up in the clofibrate saga. Michael writes, "We estimated that clofibrate would reduce raised plasma cholesterol by about 15%, and that such a reduction might be associated with a 30% reduction in coronary heart disease. This turned out to be a serious miscalculation. A mean reduction of 9% in serum cholesterol was achieved in the clofibrate treated group. This was less than the 15% expected and seriously undermined the prediction of the CHD reduction. The incidence of non-fatal myocardial infarction was 25% lower in the clofibrate group compared with the high cholesterol control group, but the incidence of fatal heart attacks and of angina was not different. Excluding vascular diseases, there was an excess deaths of 77 deaths versus 47, due principally to biliary and intestinal causes in the clofibrate treated group, and an excess of gallstones and cholecystectomies. So activating PPAR alpha did not reduce fatal heart attacks, but it did increase deaths due to bile acid issues. One of the things that makes biology eternally fascinating, but sometimes frustrating, is that biological systems like to do something I call creative reuse. Many of these pathways are used in several different processes, or many different processes. In fact, this is not the way a human would design a system. It says in this paper, the nuclear receptor PPAR alpha is known primarily as a regulator of fatty acid metabolism, energy balance, and inflammation. But evidence suggests a wider role in regulating the biotransformation of drugs and other lipophilic chemicals. So PPAR alpha, as it's regulating fat metabolism, is also involved with detoxification. It's hard to imagine why biology would combine lipid metabolism and detoxification, but I'm going to put forth my hypothesis as to why that might be, how and why this works in real organisms, why this presumably also happens in humans. So this paper actually shows how PPAR alpha is a transcriptional regulator for detoxification of plant diet-derived unfavorable compounds. So PPAR alpha sits right here, and this thing, phytanic acid, is actually a branched fatty acid, which is actually found in plant chloroplasts. And phytanic acid on its own is actually toxic to animals. And so phytanic acid is a ligand of PPAR alpha, and that means that it activates it, as do polyunsaturated fats. So you've got polyunsaturated fats and branched fatty acids, both activating PPAR alpha. And one of the things that this does is it activates peroxisomes, and that's why PPAR is a peroxisome proliferator, and that's hence the PP. And so PPAR alpha activates these pathways that activates peroxisomes, and it sends these branched fatty acids to the peroxisome to get essentially shredded. What the peroxisomes do is they break fatty acids down into acetyl-CoA, and fats are made of long chains of carbon. They can be anywhere from 4 up to 26 carbons long or longer. I've seen as long as 32. And the reason the lengths of the fats are usually in multiples of 2 is that they're built by these individual units of acetyl groups, which are just little 2 carbon groups. And so you can, if you put 4 acetyl groups together, you end up with an 8 carbon long fat. And so fats are built in the cytosol of the cell, the main part of the cell, but they're broken down either in the mitochondria, where it breaks them down into acetyl-CoA, and then the mitochondria uses that acetyl-CoA and it oxidizes that all the way down to create ATP, or fats are sent to peroxisomes. And what the peroxisomes do is they break down the fats into acetyl groups, and then those acetyl groups are exported, and they can either go into the mitochondria to get burned to make ATP, or they can go into the cytoplasm and they can actually be used for synthesis of other fats, or used for synthesis of things like cholesterol and bile acids. And when we talk about detoxifying, we can think about xenobiotic metabolism. Basically means the metabolism of drugs, which are not things that are normally found in nature, but you give a drug to someone, it goes to the liver, of course, where it's broken down by enzymes called cytochrome P450s. The enzymes that control this are the aryl hydrocarbon receptor, PPAR alpha, and CAR, which is another nuclear receptor. These things are all nuclear receptors. They're molecules that hang out in the cell, and they look for specific compounds. And when they get activated by those compounds, in the case of PPAR alpha, it's things like polyunsaturated fats and branched chain fats. When it sees those things, it starts to create these cytochrome P450s and these other enzymes. And all of these enzymes are involved with detoxification of fats, of drugs. When those drugs get broken down, they get excreted either in the bile or in the urine or in the feces. And interestingly, this is all wrapped into circadian rhythms, and they call this circadian detoxification in this paper. And so PPAR alpha is somehow wrapped up in fat metabolism, detoxification, and circadian rhythms. And to show how important this detoxification process is, in these mice, they fed them sesame seeds, just sesame seeds. And this is these are normal mice here. And this is actually a survival curve. So what happens is so at the beginning, there's 100% of the mice surviving. The wild type mice, just normal mice, this is 100 days long. 100% of normal mice survive the 100 days. Now, the mice in blue don't have the gene PPAR alpha. And if you feed mice that don't have PPAR alpha, the sesame seeds kill them quite quickly. You can see here by day 10, roughly 75% of the mice are dead, poisoned by sesame seeds, which obviously does not poison mice with a PPAR alpha gene. Here's another example of this. The mice who didn't have the PPAR alpha were fed fish oil in this case. And you can see that by day 12, 80% of the mice were killed by fish oil due to acute liver failure and acute liver toxicity. And so, in fact, these detoxifying enzymes created by PPAR alpha are incredibly important for the survival of mammals who can't handle having too many of these very unsaturated omega-3 fats in their livers. And I think these processes are really difficult to understand, even for professional scientists in the field. It's really hard to wrap your head around why circadian rhythms, inflammation, lipid metabolism, and detoxification should all be sort of lumped together into one pathway, one enzymatic pathway or one transcriptional pathway that can be activated by something like olive oil or fish oil. And that's, of course, why I think it's really important to always keep an evolutionary mindset. You have to put the problem into the perspective of evolution, and you usually have to think way back. So the ancestors of mammals and the ancestors of earthworms diverged over 500 million years ago, it seems. Earthworms have really weird fat composition compared to mammals. And so this is an earthworm, and you can see there's two columns here. This column is very long because earthworms have all of these weird fats that you don't see a lot of many places. And you see they caught earthworms in the spring and they caught earthworms in the winter. And so here's the saturated fats. Saturated fats are pretty straightforward. You again see the seasonal variation in the amount of saturation of fat. So it's 33% saturated in the spring, and they're only 19% saturated in the winter. And I've shown before the same thing in ants. Ants are more saturated in the summer and the active season than they are in the winter. Things like acorns and pine nuts are another example of very unsaturated fats that are seasonally available in the fall. And so you have very highly unsaturated fats available in the times when animals are becoming torpid and when they're going into hibernation. One of the weird things about earthworms is they have very low monounsaturated fat levels. So you can see there in the spring, add those numbers up, it's like 7% monounsaturated fat. But what they have is they have tons of long chain polyunsaturated fat. And so they're almost a little like fish, sort of, except they have an almost even amount of omega-6 and omega-3 fat. So this 25 omega-3, this is EPA, we find this in salmon and fish oil, and you can see that's almost 13% of their fat in the spring and over 14% in the winter. And this one, 24 omega-6, that's arachidonic acid that's made from linoleic acid through the enzymes delta-6 desaturase and delta-5 desaturase and others, elongases. And I've talked a lot about D6D and D5D in past videos and how they predict bad outcomes in humans, things like diabetes. And so the earthworms have a tremendous amount of unsaturated fats, and especially of these polyunsaturated fats. And so that's about, so the unsaturated fats are 53% of their fat in the spring, but it rises to almost 70% in the winter. But the other thing the worms have is they have about 10% of these branched fats. These are all, there's a whole bunch of different ones. But they all activate PPAR alpha. And as you remember, the polyunsaturated fats and branched chain fats both activate PPAR alpha. And so you could imagine that the fat of earthworms would be very activating of PPAR alpha. And the other interesting thing is mammals, the fat in mammals is overwhelmingly composed of 4 fats. That's palmitic acid, which is the 16 carbon saturated fat. 18 carbon saturated fat is stearic acid. 18.1 is oleic acid, like in olive oil. And 18.2 is linoleic acid, like in corn oil. And so in worms, those 4 fats only make 21% of the fat in the worm. We believe that the first mammals ate insects. These were small mammals. They lived around the same time as the earliest dinosaurs, or perhaps before, and they were eating, like I say, mostly insects. And so there are still mammals around like this. This Sorex araneus is the common shrew, and shrews live underground. They have a very high metabolic rate, and they eat mostly worms, bugs and worms. And so you can see this, these are worms right here. And this is sort of like an estimation of volume. People went around and they caught all these shrews, and they looked at their stomach contents and at their intestinal contents to try to figure out what shrews eat. And so this first column, I summed it up, and it's about 43% of their diet is earthworms. And yet the body fat of the shrews is not very similar to the earthworms at all. You see they have a lot of these weird fats because they're right. If they're eating worms all the time, they're constantly taking in all those branch chain and all the weird things. And so they do have a little bit of them, but compared to the earthworms, they're very minor components. And so like in all mammals, when you look at the total of palmitic, stearic, oleic, and linoleic acids, in the shrews, these are three different organs. This is liver, this is brown fat, and this is thigh muscle. But in every case, the body fat of the shrews is 63 to 74%. You know, the big four, right? Whereas the earthworms are only 21%, the big four. And so that suggests to me that by some process, the shrews are actually converting those weird unsaturated and branch fats into fats that are more appropriate for mammals. And so this is in rats. This is not actually in shrews, but rats and shrews both presumably have a lot of peroxisomes. And like I said, what the peroxisomes do, they don't make ATP. They sort of do thermogenesis. They create heat and they create hydrogen peroxide, and they break these long chain weirder fats into acetyl-CoA groups that the organism can use for its purposes. And so they fed these rats different fats, and they looked at its effects on, this is palmitoyl-CoA, so this is palmitic acid, and how quickly is it oxidized in the mitochondria or in the peroxisomes when they fed the rats different fats. And so palm oil is the baseline. That's it's about half saturated. Palm oil is, and then they move to safflower oil, and you can see in the mitochondria, there's not much difference between omega-6 linoleic acid and saturated fat. It's about the same. There is a slight increase perhaps with linoleic acid and the amount of peroxisomal oxidation of fats. But perilla oil is an omega-3 fat. That's alpha-linolenic acid. And so you can see with the perilla oil, peroxisomal oxidation doubles. It goes from 2.6 all the way up to 5.5. Mitochondrial oxidation also increases by around 50%. And then they try fish oil. And fish oil, peroxisomal oxidation really dramatically increases all the way up to 9.25. So that's more than a tripling of the ability of peroxisomes to break down fats when the rats are fed fish oil. And again, when I say oxidation, what I mean is breaking that fish oil into acetyl-CoA groups. Peroxisomes are not mitochondria. They're not burning that acetyl-CoA to produce ATP. They're just taking these long, weird fats and they're just breaking them down. They're literally sending them to the shredder, right? The peroxisome is the shredder. So the rats, when they eat a bunch of fish oil, or if they're a shrew eating a bunch of branch chain fats, what happens is they're going to activate peroxisomes and they're going to send those omega-3 fats and those branch chain fats into the peroxisomes to break them down into acetyl-CoA groups. And you can see this is just a study pointing this out, that branch chain fatty acids are mainly degraded in peroxisomes in the rat liver. Branch chain fatty acids cannot be transported across the mitochondrial membranes. They are directed to peroxisomes for beta oxidation. And beta oxidation is the process of shredding fats into acetyl-CoA groups. And so fenofibrate is a drug that activates PPAR alpha. And you see that just exactly what I'm saying. The fenofibrate, when you activate PPAR alpha, you do CPT1A, controls how fast fat goes into the mitochondria, and it does go up by about 40%, and that increases mitochondrial oxidation of fats. But AOX is an oxidant enzyme that lives in peroxisomes, and you can see that goes up by 4.5-fold when you use fenofibrate and you activate PPAR alpha. And so when you activate PPAR alpha, you massively increase the activity of peroxisomes. And I have peroxisome oxidation here. It is being oxidized, but you could also just say peroxidome shredding. And so both in the mitochondria and in the peroxisomes, we're shredding those PUFA. Furthermore, when you activate fenofibrate, when you activate PPAR alpha, you upregulate all these genes that are in charge of making monounsaturated fat. You can see SCD1 is up 2.6 fold. Fatty acid synthase is up 50%. All of these are these E-love L5 elongate fats and turn palmitic acid into stearic acid so that it can then be desaturated by SCD1 into oleic acid. And furthermore, the PPAR alpha upregulates delta-6 desaturase, and delta-6 desaturase converts things like alpha-linolenic acid to the long-chain omega-3 fats. And so when you saw that perilla oil that was full of alpha-linolenic acid, which is the plant-based omega-3, that activates PPAR alpha, and the body is just like, we are going to use every tool at our disposal to get rid of this alpha-linolenic acid, right? We're going to send it into the peroxisomes, break it into acetyl-CoA, we're going to send it into the mitochondria, and we are going to desaturate it and try to turn it into DHA, and then we're going to rebuild it into oleic acid. That is absolutely the plan. And remember that the polyunsaturated fats themselves and the branched chain fats are the things that in normal biology would activate PPAR alpha, and they're also the toxins. I showed the experiment where mice that don't have PPAR alpha, if you feed them enough fish oil, it kills them because of acute liver failure. And so when you start to roll this all together, you can, all right, so circadian rhythms, detoxification, fat metabolism, why shouldn't they be controlled by the same pathway? Those polyunsaturated fats and those branched chain fats coming in when they're maximal is a signal that winter is approaching. You have to change your fat metabolism to go into torpor and to get ready to hibernate, but you have to absolutely get rid of the PUFA, right? Or you'll have acute liver failure. Anyway, and so just to show that, this is mice treated with fenofibrate, and this time they looked at the actual fat profiles, and I've showed this before, but we're going to look at a slightly different light here. And so they give these mice fenofibrate, and look what happens to, this is linoleic acid, right? So this is an omega-6 PUFA, and in normal mice, it's at 1100. You give them fenofibrate, right? Peroxisomes are increased. Beta oxidation of mitochondria is increased. Delta-6 desaturase is increased to make it longer things, and it gets sliced in half, right? We're shredding PUFA. PUFA comes with its own signal, please shred me, right? Get rid of me. This is 22, 6, and 3. This is DHA. You can see DHA also goes from 190 down to 140. This is alpha-linolenic acid. Alpha-linolenic acid is never high in tissues because the body tries so hard to get rid of it. But even that goes from 40 down to 13 with fenofibrate. And at the same time, here's our oleic acid, our MUFA, and that's going from 800 up to 1200. And so the body, and also saturated fats drop. So stearic acid goes from 350 down to 250 because SCD1 is activated by PPAR alpha, right? And SCD1 converts stearic acid, saturated fat to oleic acid, monounsaturated fat. And so when you activate that PPAR alpha, you are absolutely shredding these PUFA and rebuilding them as MUFA. This is just a quote from that same study saying, our data confirm the massive liver fatty acid beta oxidation induced by the pharmacological activation of PPAR alpha results in a tissue deficiency of essential fatty acids. So when you activate PPAR alpha, PUFA levels drop. So this is a study with clofibrate in humans done way back in 1980, showing that same phenomenon. These are the people at the beginning of the study, and for 2 months they're told to eat a lot of polyunsaturated fat. They're told to eat corn oil, they're told to eat soybean oil, they're told to eat margarine, and so they go out and they eat a bunch of polyunsaturated fat, and you can see that linoleic acid does indeed go up, and this is in their phospholipid membranes. So I've talked a lot about the omega quant test. This is essentially the same test where they're testing their phospholipid membranes, and you can see linoleic acid goes up when they eat the PUFA. And then after 2 months of that, they start taking clofibrate. And after 4 months of that, you see the PUFA goes back down, despite the fact that they're still being told to eat lots and lots of PUFA. But the drug reverses that, and it also increases MUFA from 13.4 up to 15.8 after they start clofibrate. You can see stearic acid drops, and if you go longer, if you go 6 months, stearic acid drops even more. At the bottom here, I put the DI-18, so that's activity level of SCD1, and you can see after they eat a bunch of vegetable oil, it's at 0.77. After they start taking clofibrate, which activates PPAR alpha, it goes up to 0.95. And after 6 months, that ratio goes up to 0.98, showing that PPAR activators do indeed increase the activity of SCD1 in humans. There are other studies showing that when humans eat alpha-linolenic acid, we absolutely shred it and rebuild it into saturated fats and oleic acid. They do this with tracers, so they take a radioactive label, and they add it to alpha-linolenic acid. People consume that alpha-linolenic acid, and they take samples. They take tissue samples from the people later and see what happened to the that radio labeled carbon from the alpha-linolenic acid. And so what happens is we break that down through beta oxidation into acetyl-CoA. And so alpha-linolenic acid prefers to go to the peroxisomes, but it can also be broken down in the mitochondria. Either way, we break it into acetyl-CoA. Some of that gets broken down to CO2. We make ATP, and it's excreted in the breath. But a lot of it gets rebuilt to palmitic acid, stearic acid, and oleic acid, just like we saw in the mice given clofibrate. This phenomenon in humans is called carbon recycling, because you're eating one fat, you're breaking it down, and you're rebuilding it into another fat. Peroxisomes are very interesting. So like I said, they don't make ATP. What they do when they're doing beta oxidation is they're really just generating heat and hydrogen peroxide. One of the functions of the peroxisomal respiratory pathway is disposal of excess reducing equivalents. And in the last video, I talked about the context of PPAR alpha and whether or not NAD plus was available, or whether or not there was too much NADH. And so one of the things peroxisomes can do is they can actually get rid of NADH. When you produce peroxide, there are enzymes that can use that peroxide to convert NADH back to NAD+. An example of this is the enzyme NNT. And even though the peroxide is produced in the peroxisome and NNT lives in mitochondria, hydrogen peroxide has the sort of unique ability to diffuse both through water and through cell membranes. And so if the peroxisomes are making a lot of hydrogen peroxide, that can move throughout the cell and into the mitochondria and help relieve a buildup of NADH. There is a huge difference between the peroxisomal activity in mice and in rodents and in humans. This might be because rats and mice eat a lot of seeds. Seeds tend to be full of polyunsaturated fat. Humans evolved from chimpanzees that eat mostly fruit. Fruit do not have much polyunsaturated fat in them, and so we might have just turned down the peroxisomal part of our metabolism because we didn't need it so much. But you can see here, if you give clofibrate to rats, their peroxisomes increase. This is an enzyme that's in peroxisomes, and you can see it goes up by 940% versus if you give it to humans, it goes up by 70%. And even the starting rate of ACOX activity in rats is much higher than in humans. And so clofibrate does indeed increase peroxisomal activity in humans. It's just not nearly as dramatic of effect in rats, and there's not as much to start with. And that might go some of the way. I'm speculating here, but I'm curious. So one of the things that we see with drugs like fenofibrate and clofibrate is that a lot of times they work well in mice. And so this is a type of mice with this inherited fatty liver disease. And this is the amount of fat after 15 weeks in these mice. But if you give them fenofibrate, the amount of fat in the liver is just massively. And so because of these results in mice, everyone is going along thinking, well, first off, these clofibrates have been used as cholesterol lowering drugs for 60 years now or 50 years now. And so they're seen as a good thing. And everybody knows because of the mouse studies, etc., that these PPAR alpha drugs can reduce liver fat. And so in 2018, they did, and I showed this in the last video, but they did a study in humans. And for the first time, they gave humans a fibrate drug, and they actually measured the amount of liver fat accumulation. And you can see in humans, there's a 25% increase in liver fat, as opposed to mice where you see this massive decrease. One thing I can say is that I am not buying the idea that PPAR alpha controls fatty acid metabolism. It sits downstream of the ARA hydrocarbon receptor. The AHR is activated by things like benzopyrene, which are cyclic molecules found in charred foods. So when humans started grilling meats and started cooking other foods, we might have been exposed to more benzopyrene. When the AHR is activated, or when we eat things that we have to deal with or otherwise that would cause liver damage, such as polyunsaturated fats, all of those things activate PPAR alpha, and that increases the activation of our peroxisomes, which are essentially giant paper shredders, but for fats, for polyunsaturated fats, for branched chain fats, and for other kinds of weird biological molecules. We send those weird biological molecules to the peroxisomes to get rid of them. We shred them down to the component parts. We can use those component parts to rebuild fats that are biologically appropriate for mammals. So what should you eat? I'm on the record as not being a fan of olive oil, certainly not if you are obese, if you are pre-diabetic, if you have metabolic issues. When you eat olive oil, you make something called OEA, which is one of the most potent activators of PPAR alpha that we have. You might also be thinking, well, maybe we should be massively activating PPAR alpha because we've probably eaten too many polyunsaturated fats and we want to get rid of them. And that sounds good, except the problem is that activating PPAR alpha increases delta-6 desaturase and SCD1. Delta-6 desaturase takes the linoleic acid that you do have, it converts them into signaling molecules, these oxylipins, that are strongly associated with obesity. SCD1 is strongly associated with obesity and lipogenesis. And so even though you're getting rid of PUFA, it's not necessarily a good thing because you wind up essentially triggering that torpid metabolism and you wind up shifting into fat storage mode because of all the oxidized polyunsaturated fats. Should you eat fish oil? Well, if you don't have a PPAR alpha gene, fish oil is apparently toxic. However, coming up soon, I'm actually going to talk about the benefits of DHA. DHA is a long-chain omega-3 fat that seems to act very differently than the other PUFA. It's kind of the final end product, the longest, most unsaturated omega-3 fat that the body keeps around. And so there seems to be some feedback mechanisms whereby having DHA specifically can help slow down a runaway delta-6 desaturase gene and can help slow down an SCD1 gene that's out of control. I'm just going to take a second and again to plug the Omega Quant test. You can test your membrane fats. Google Omega Quant complete test. Use my discount code FIRE, F-I-R-E. You will save 10% off that test. They'll send you out a test kit. It takes a few minutes to put a blood spot on there, send it back to them, and you can get a full readout of all of the fats in your phospholipid membranes. This is super useful to tracking how you're doing. Are your desaturase indexes going up? Are they going down? What about SCD1 levels? You can share that info over on the Reddit group r/saturated fat. There's a great discussion group. Lots of people have put their Omega Quant test there. Join in the discussion. Thank you guys. That's all I got right now. Come back next week. See you then.